In 326 BC, Alexander the Great was marching his armies across the known world, carving out a massive global empire through military force. Back in Greece, his former teacher, Aristotle, was engaged in a different type of conquest. He wanted to map the boundaries of the natural world. Known today as the father of biology, Aristotle drew the first lines of scientific classification, dividing the animal kingdom into two basic groups, those that lived on land and those that lived in the water. It was a simple start, but it established a practice that has continued for centuries, grouping and labeling nature to find a predictable order in the environment. This desire for order eventually led Carolus Linnaeus to formalize the binomial system, a universal rulebook that assigned a two-word Latin name to every living thing. Under this system, common wheat became Triticum aestivum, and the tiger became Panthera tigris. This image outlines the classification of our own species. Within this biological catalog, humanity is assigned the label Homo sapiens. While these categories organized life from the outside, the actual mechanism controlling human development was hiding deep inside ourselves, operating by a set of rules no one could see. To find those rules, we have to look past the organs, past through the cell membrane, and examine the microscopic landscape of human genetics. Down here, the blueprint for a healthy Homo sapiens relies on mathematical precision, exactly 46 chromosomes, arranged in 23 pairs. The first 22 pairs build our general physical traits. The 23rd pair determines biological sex, configuring as either XX or XY. When all 46 threads are present and intact, the body operates as a balanced biological machine. But this balance is fragile, leading to the question, what happens when nature makes a microscopic typo? Any alteration to this blueprint, whether a change in the total number or a structural defect, results in chromosomal syndromes. These are incurable conditions written directly into the body's code. If the 23rd pair is missing one of its X chromosomes, the total count drops to 45. These notes detail the result, Turner syndrome. It affects females and disrupts physical development, often leading to incomplete organ growth and permanent sterility. Conversely, adding an extra chromosome to that sex pair, bringing the total to 47, results in conditions like Klinefelter syndrome or Jacob syndrome. A single missing or added microscopic thread permanently alters the physical and developmental path of the individual. These errors occur outside the sex chromosomes as well. When an extra copy attaches to one of the standard pairs, it creates a condition known as a trisomy. This diagram outlines trisomy 21, or Down syndrome. That single extra chromosome of the 21st pair impacts physical features and cognitive development. Similar numerical errors at other locations produce severe physical defects, like the cleft palates and heart conditions found in Patau syndrome at the 13th pair, or Edwards syndrome at the 18th. The total count is only part of the equation. The physical shape of each individual chromosome must be perfect, too. If just the upper arm of a chromosome on the 5th pair breaks off, it causes Cre-Du-Chat syndrome, leading to low birth weight and distinct developmental issues. The margin for error is effectively zero. Even a fractured piece of one microscopic thread results in a profound, incurable condition. When we finally pull back from the microscopic cellular level and look at a complete living person, we see how the focus of biology has shifted over time. Aristotle began by grouping the massive animals walking the earth. Today, we trace lifelong medical conditions down to a damaged arm on the 5th chromosome pair. Studying this science requires looking past historical dates and Latin names. It is the study of how precisely our biology must be configured to function. Our physical development and our health operate on this precise balance, anchored to exactly 46 tiny threads.